our next session coming up is on, let me pull this up, is on what does the future look like? So essentially, we're going to talk about what treatments might be available short-term and long-term that are on the way for a cure for type 1 diabetes. We're going to join citizen scientist Karen Jordan, who I'll introduce in just a moment. And our renowned panelists, we have Dr. Matthias Hebrock, an expert in generating stem cell-derived beta cells, Dr. Jeffrey Bluestone, one of the world's leading immunologists, and Dr. Meyer, who has developed a protocol to transplant kidneys without requiring long-term immunosuppression, which is very cool. They're going to describe their visions of what the future will look like and how their work that they're doing today is actually contributing to progress we're making in cure research. So for some general housekeeping, again, thank you to all of you who were joining us for the last panel. Thank you to those of you who've just logged on for this one. We look like we maybe have about 20 new folks who've joined us, so thank you and welcome. My name is Melissa Lee, and I was moderator for the last chat, and I'm going to help us kick off here. So again, we encourage you to ask your questions with the Q&A feature. And those can be, we'll get to those as we can at the end of each session. And so we look forward to hearing you for that. I am now going to introduce your moderator for your next panel. This is Bay Area citizen scientist Karen Jordan, and she will be the on-screen panel facilitator and the moderator of all of your Q&A as the questions come in. And I'm sure you're going to have a lot of questions about this one because it's all very exciting research. So let me briefly give a bio on you, Karen. Thank you for joining us today. Karen became involved with the JDRF when her daughter was diagnosed in 2008. She was a member of the International Board of Directors for JDRF for 3 years, and she's currently serving on the T1D Fund Board, and she's also on the Research and the Audit and Risk Committees for the JDRF. So thank you for great involvement in all those years. Let's see, immediate past GBA chapter board president, nominating and board development committee member, and lead volunteer for JDRF Northern California Center of Excellence at Stanford and at UCSF. If you've been to any Bay Area events over the years, you have likely seen Karen there. I know I've seen her many, many times. She's a founding board member of CarbDM, who's one of our hosts for today as well. And let's see, lots of different nonprofit work that I'll save a lot of this, but essentially we are so fortunate to have Karen with us today, and I will kick it off to you, Ms. Jordan.
Thank you so much, Melissa. I really appreciate the kind introduction, and welcome to everyone who's attending the summit. Really appreciate you joining us on this Sunday from literally around the world. We are so lucky to have 3 world-class scientists with us for the next hour to discuss the state of research focused on cures for type 1. So I'm just going to ask each of them to introduce themselves before we kick it off, and we will go in alphabetical order. So just to clarify, that means Dr. Bluestone first, Dr. Hebrock second, and Dr. Meyer third. So Dr. Bluestone.
Great. Well, thanks, Karen, and thanks to JDRF for organizing this. I think it's a great opportunity to talk about where we are in the field right now and where I hope we're going. Can people hear me okay? Is my volume okay? Good. So my background, I'm a card-carrying immunologist for over 30 years, working in the field of immune tolerance and trying to understand the immune system and how to control it, in particular in type 1 diabetes, which is a clear and present danger in our autoimmune sphere. I've been involved in a number of different initiatives, both aligned with the JDRF, having been head of JDRF centers over the years, as well as with other government and university organizations, most recently the head of the Diabetes Center at UCSF before my current positions outside UCSF now as CEO of Sonoma Biotherapeutics, which is interested in developing cell therapy for the treatment of type 1 diabetes. And as you'll hear, I've been involved in a number of drug areas, including the development of teplizumab, a drug that hopefully will make it to the finish line in the future.
Thank you. Dr. Haybrock?
Good morning, everyone. It's great to be with you virtually. I see we have almost 200 participants. That's fantastic. So I've been part of these diabetes summits for a number of years now, and I always think there's a great energy. So hopefully we're going to get this across through the ether today as well. So I am currently the director of the Diabetes Center at the University of California, San Francisco. I took over when Jeff took on a new position almost, I think now 10 years ago. By training, I was trained in something that we call developmental biology. That's just a fancy term for us trying to understand how organs and tissues are being generated and how they develop during embryogenesis. But the overall goal always was, and that's what we have been doing for the last several years, is to use that information about how things are being put together under the normal circumstances to rebuild stuff that we would like to have from embryonic stem cells. In our case, of course, these include the insulin-producing beta cells that we're now generating from human cells. I've been involved with a number of companies in this particular area, most notably and most recently Semma Therapeutics as well as ViaCyte a couple of years ago. And therefore, I've been in the space of cell therapy for a long period of time.
Thank you. And then Dr. Meyer.
Hi, yeah, I'm Dr. Meyer. I'm a practicing physician scientist. Expertise is in blood and marrow transplantation and immune tolerance. It's incredible to share the stage with Dr. Bluestone, who is inspirational, I think, in the field, and actually has been a large part of his work has been a large part of re-identifying into T1D research that has occurred in the last 4 years in my career. I am part of a large team that is focused on methods for inducing immune tolerance in clinical practice. I did not invent the tolerance mechanism at Stanford that we're hoping to pioneer and deploy in islet transplantation and potentially eventually in new onset disease, among a number of different potential applications.
Thank you. So as you can see, we're going to have some tremendous discussion based on the experts who are participating this morning. Just to highlight the fact that there's been tremendous progress both on the cures front, which we'll get into, but also on the treatment front, which JDRF calls the improving lives side of the equation. And I just wanted to briefly mention that there's so much incredible work going on improving the insulins that are available, work in the ultra-rapid insulin space, the glucose-responsive insulins, the co-formulated insulins, for example, with pramlintide, which is another hormone, and insulin, and also liver-targeted insulins. You know, I think many of you are benefiting from the hybrid closed loop systems, 2 of which are approved now, and there are several others that will be coming onto the market and available for patients very soon. And then also the adjunct therapies, which are therapies in addition to insulin, some of which are approved already in Europe, and some of which are in clinical trials, both here and abroad. So really exciting progress on helping those with type 1 manage their glucose levels. But I think equally and probably more exciting, at least to me, progress on the cures front. So let me just try to frame this up a little bit before we get into the questions and the discussion. We know how to cure this disease. The concept has already been proven. There are people who have had islet transplants who are walking around without having to take exogenous insulin for 10 plus years. And it's remarkable, and it's pretty much pointing to the basic fact that we know we need to replace the beta cells that are missing, and then we have to kind of fix the autoimmune response. So why is this not available for everyone? 2 problems. One is that there is just not enough of a supply of islets to transplant. And secondly, those who receive islet transplants need to be on immunosuppressants for the rest of their lives. So not really a viable option for most people with type 1 diabetes. And you will hear that our panelists are working on both sides of that equation to actually address both issues that come along with type 1 diabetes, and get us to a cure. So, you know, as you heard, we have the perfect panel for this discussion. We've got immunologists, we've got stem cell biologists, and so we will get into their work shortly. But I just wanted to kick off. I know that when everyone hears about cure panels, they all want to know, you know, when is it coming? What's going to happen? And kind of what's the pathway to getting us to that full biological cure? And, you know, I know that we're not going to get from where we are today to kind of the ultimate cure overnight. It's going to be a progression. And we're going to see steps along the way to getting us to what that ultimate goal is. So I was just going to throw out to each of you, you know, what does that pathway look like? And are you comfortable putting some kind of general timeframes around short-term and long-term? And whoever wants to go first.
Well, I'm happy to start. I maybe look at this a little bit differently in the following way. The notion that we are going to one day go from not being able to treat or cure this disease to being able to cure this disease is an unlikely scenario to me. I think the more likely scenario is the scenario that's happening in cancer now, where for many decades, what we were able to do is develop drugs that prolong people's lives, which was great, but didn't cure people. And I think what's changing now in type 1 diabetes is that we have lots of ways of helping patients manage their disease, helping patients live a much longer and healthier life. But the cures are few and far between and at a cost. You gave the example of islet transplant. So for me, I think the first things we're going to see are that a subset of patients, a small subset, maybe based on a combination of where in the disease progression they are, where in the genetics and other subtypes of disease they have, that these patients will come off of insulin. It may be initially 10%, 15%, not unlike what happened in cancer immunotherapy when we finally figured out the new generation of immune drugs that help to fight cancer. I think we'll have a new generation of immune drugs which will get a small subset of people initially off. But that's going to be transformational. It'll be transformational because once you know what great looks like, once you know what a cure looks like, then you can build from that. So instead of just shifting the curve this way, all of a sudden you can start shifting the curve up and moving so that if this year it was 10% of the patients, then maybe in 5 years it's 30%. And so I think the way this is going to happen is incrementally. I think it's going to happen for some people sooner than others. In the short term, I think you're going to see a subset of people that actually can be cured from this disease, not unlike what happens with islet transplant. And then over time, we will increase those numbers.
So if I ever just briefly chime in here, and I'm also coming, there are different ways of defining a cure, of course. For me, a cure, and I give you the definition I have, just having had my coffee, is that you wake up in the morning and you have a cup of coffee and you really don't care if you put sugar in there or milk in there or anything else, and you just have it. You don't have to worry about what you take into your body. So for us to get to a cure, it's a really big word, but I think we have, as Jeff was saying, made a tremendous amount of incremental increases already and advantages already, and we are essentially putting the building blocks of what a cure would look like together right now. So that what you need, and Karen just said this, is if you strip everything away, the one cell that is destroyed in the body of patients with type 1 diabetes is the beta cell. We cannot give everyone islets right now. We just don't have enough of those because we're taking them out of, unfortunately, people who have died, often through accidents. So cadaveric islets are just not going to be enough to treat everyone. But by us and others and companies are now being able to generate cells that look similar, if not completely identical to the beta cells that we have in our body, the supply problem is going to go away. I'm more than happy to acknowledge that there's a number of other issues, of course, with immunology, but we're learning so much on that side as well. So I think Jeff is right. We're not going to be able to treat everyone on Monday in a year from now, But I think we're learning so much about how to accelerate our work, how to protect these cells that we would like to put in there, how to modulate the immune system in a way that when we get ready, I think hopefully we can ramp up faster and sooner than later.
So I would echo the comments that are made. I think that I work in cancer immunotherapy as well, and I agree completely with Dr. Bluestone. This is going to progress in pockets over time as we learn more and cure subsets of patients. For example, with islet transplantation, we have a limited supply of islets, but we can learn a lot, and we can hopefully get patients in the next 5 or 10 years down to minimal drugs or even potentially off drugs as proof of concepts. I think what is also very exciting is if you think about the clinical infrastructure and just what a hospital looks like today, it's starting to shift. I mean, where are patients in the future who are going to receive cures treated? Right now, the infrastructure doesn't exist very substantially. I think over the next 5 years, you're going to see a lot of practitioners, multidisciplinary teams come together around clinical trials in particular, but eventually around standard of care to just offer this to more patients more readily in a concerted effort.
Thank you. And maybe to follow on that, if you each wouldn't mind going through in a little more detail about the research that you are all focused on. You know, as I said, we've got the beta cell side represented. We also have the immune side represented. So maybe if Dr. Haybrock, you want to start with talking a little bit more about your work with stem cell derived beta cells.
And I would be remiss by not mentioning that we are part of a team, of course. We're not just working in isolation. And one of the biggest teams that I think we recently put together actually does include both of my other panel members, Everett as well as Jeff. We're part of what is now supported by the JDRF. It's what we call the Northern California Center of Excellence, in which we try to combine exactly what we're talking about today, the strengths that we have at UCSF as well as at Stanford, in trying to push these things forward in terms of towards a cure using stem cells. So what I would like to point out is that a couple of things have happened over the last several years that just, I think, give me and hopefully many other people hope. And that has to do with that we, as I mentioned already, now being able to generate cells almost at will from human embryonic stem cells or from induced pluripotent stem cells. What that means is that we can choose what kind of cell we can generate. So for example, in the context of the beta cell, it often sits together with other cell types as well. We call this a human islet, and we learn more and more about how specific cell types in this islet actually talk to each other and work together. And so we can now generate structures that look like human islets and then have embedded beta cells that now sit in the environment that they normally sit in your body. And I think that is going to be very helpful. Second thing that has happened recently over the last several years is just a revolution in what we call gene editing. Gene editing, again, is just a fancy term for us being able to go in almost like a mechanic into an engine and just modify parts of cells at will. To give you an example, we have 3 billion base pairs. That's essentially in every single cell. That's 10 times more as the population that we have in the United States. With these tools, and many of you guys might have heard about this, they're called CRISPR, we can now go into any given cell and modulate, manipulate, take out, add just one of those 3 billion base pairs, or 1,000, or even more. What that allows us to do is now to build beta cells that we believe are just going to be more advanced compared to what we had in the past. What I mean by that is, even if I give you a beta cell today, if I generate a beta cell out of a stem cell and I give it to a patient who has diabetes, unfortunately, the immune system is revved up to destroy exactly that cell that is needed, the beta cell. That's how you got to type 1 diabetes in the first place. The immune system has decided to treat the beta cells in your body as foreign. So if I give you more of those beta cells, the immune system will again decide that these are cells that do not belong to you and will destroy them. Now with this CRISPR technology, we are doing this in the lab, and again, several others around the world are doing this as well. We now can go in and we can modify molecules that are being presented to the immune system by the beta cell. For those of you guys who have younger children, it's the cloak of invisibility that Harry Potter had that we would like to generate for our beta cells. Beta cells that are in stealth mode, that they can sit in your body and not being recognized by the immune system. And I don't want to glance over all of the problems that come with it. That's not going to be easy to do all of this. We have come quite a way already in generating these kind of cells, but there's lots of stuff that needs to be addressed and needs to be investigated. And that's why being able to be in the Bay Area with friends and colleagues like Jeff and Everett and others in this Center of Excellence, we have beta cell stem cell biologists talking to immunologists all the time. And this intersection of talking to each other and therefore coming up with strategies that help the beta cell minimize the risk from the immune system, that I think is how we get to the cure.
Thanks, Matthias. And thank you also for mentioning the JDRF Northern California Center of Excellence. I think it's going to be a transformative effort, and it involves 13 of the top scientists at UCSF and Stanford who are working collaboratively, bringing together, again, as Matthias mentioned, both sides of the cure equation. So I appreciate you highlighting that. Maybe if we can go to Dr. Bluestone to talk, since you are the immunologist and maybe focusing on some of your Treg work and the fact that you've been in this work in the trenches in this work for decades.
Sure, thanks. And Everett is as much of an immunologist as I am. He's just way too shy to point that out. The way, and I think a lot of people on this webinar appreciate this, is that the immune system is this incredibly balanced setup, right? So think about a seesaw, right? When you were a kid and you're on the seesaw, the hardest thing to do was not to go up or go down. It was to stay even, right? And make sure that one person wasn't weighing a little bit too much or the other one too much. And so we learned the immune system is about that balance. If you have too much activity on one side, then you get terrible inflammation and uncontrolled autoimmune disease. And it's part of the pathology of everything from COVID-19 to type 1 diabetes, this uncontrolled immune system. And on the other side, if you have too little control, then you end up not being too little immunity. You end up not being able to control viruses or cancers when you get them. So the whole goal of immunology has always been to understand enough about each side of the seesaw that you can keep it in balance. And in type 1 diabetes, clearly the balance got off and the immune system started destroying the islets, as Matthias said. So my work, and I think a lot in the field over the past 20 years, is how do we reinstate that balance? And there's two ways to do it. One is you can eliminate some of the bad cells that are out there trying to destroy the beta cells. And we can talk a little bit about drugs that do that, like the teplizumab drug that I've been working on for longer than I'd like to talk about. Or you can reinstate that balance. You can even out the seesaw by putting more of these regulatory pathways in place to get the balance back in check. And so it's that latter area that both Everett and I work in, where we're trying to use a very specialized subset of white blood cells that circulate in everybody, that are designed to be policemen, to look when the immune system has gotten out of control and put it back into control. And these cells, which are called regulatory T cells or Tregs, are really a fantastic machine for delivering molecules, proteins, factors that will keep the immune system in check when you want it. And the question that we're all trying to work on in this field right now is, can we harness that? Can we isolate those cells from the body? Can we use the tools that Matthias is talking about, these engineering tools, to make these cells bigger, better, more potent, and then put them back into patients and let them go to the pancreas and reestablish that immune balance that we're talking about? So on the one hand, as an immunologist, we're trying to get rid of the bad guys that are causing the destruction. And the other is to build up on the good guys to keep the unwanted destruction in check. And I think almost every drug, almost every research project is designed to try to create that balance. Because if you go too much to one side, you get collateral damage. If you get too far to the other side, you end up being immunosuppressed to the point that Karen talked about, that really is not a long-term acceptable kind of therapy for a patient with type 1 diabetes, namely immunosuppressive drugs that you take for the rest of your life. So that's the balancing act that my research, Everett, and a lot of other people are trying to achieve.
Thanks. And Everett, I actually wanted to have you be the last in the lineup on this question, because your work is dealing with both sides, with stem cells and your work with the bone marrow, and also just your protocol that you're trying to apply towards islet transplantation. So maybe you can talk about that a little bit more.
Sure. Yeah, I think one of the major themes of the next decade is, as was described, instead of coming up with methods to break the immune system and slip islets in and then use immunosuppressive medications to control a response against those islets. The future is re-educating or reprogramming the immune system so that when you transplant new islets in, it's accepted. And there are different mechanisms that can occur, whereby that can occur. With Dr. Bluestone, Jeff's work in T regulatory cells, there's a lot of potency there, and we're exploring that. But we're also exploring the idea of creating an immune environment in the transplant setting where you take the donor islets, but also part of the immune system, the blood-forming cells, and you co-transplant them. And we've known since the 1940s that that approach can induce re-education or immune tolerance to transplanted tissues. And then, over a lot of time and a lot of work at Stanford and other centers, we've managed to actually put this into clinical practice with kidney transplant patients who receive a kidney transplant, and then also from the same donor, parts of their immune system, the blood-forming cells, and also some key immune cells. In that process, these patients can get off drugs. Not all of them, as was talked before, but 80% of them in certain contexts with genetic match. And now we're pushing that barrier further to see how we can re-educate the immune system. A major problem is that process has historically been prohibitively toxic, but there are a number of methods, including the use of T regulatory cells or specialized new medicines, antibodies that can help clear out the bone marrow, that are much less toxic. So there are ways to do this that were inconceivable even 5 years ago.
Really exciting stuff. Thank you. So Dr. Bluestone, you've talked about teplizumab a couple times. Maybe if you could talk about it a little bit more. I know the research world and the patient world was pretty excited about the announcement about teplizumab last year. So maybe you can tell us a little more about it and how it works and why it's so revolutionary.
Yeah, of course. I love talking about it. Disclaimer, I'm on the board of the company that's trying to develop the drug, and I obviously have a financial interest in it, so everybody should be aware of that. This is a drug that literally I developed in 1987. We can get into another long discussion about something that's 30 years old. It's taken so long to hopefully get to the finish line. But it was a drug that was developed out of the field of immunosuppression that we've been talking about. It was the parent drug was being used in organ transplant, but had a lot of toxicity. And we were able to develop a daughter of OKT3, which was the drug then, that provided a lot of the immune modulation without the toxicity. And basically what it does is what I had mentioned earlier. It has this unique ability when this monoclonal antibody that's against this protein on a T cell called CD3, that when this antibody reacts with that protein on an activated T cell, one that's just poised to destroy its target, those cells die. They explode. They what we call apoptosis. It's a great word, apoptosis, because you know what it means just when you say it. The cells just go pop. And what's great about the antibody, and there's going to be a class of molecules like this. is that the cells that are most poised to kill are the ones that are most susceptible to the drug. Other cells in the body, memory cells that are around because you want to react with a virus that you had 10 years ago, or naive cells that are there for when you do see a new virus. Those cells are largely unaffected. And most importantly, the regulatory T cells are enhanced by this drug. So here you have this drug that even though it hits a protein on all T cells, only those T cells that are highly activated die. And so when you treat a patient with teplizumab, you see this short-term loss of activated T cells that coincides with a rebound in the ability of islets to make insulin. And so if you have cells that can make insulin still, then you have the chance of putting the brakes on the immune system. And since it causes cell death, you can have a long-term effect even though you have a short-term treatment. So we treat for 2 weeks with the drug, and that's it. And then 2 years later, we still see effects of this drug. And the study that Karen's referring to was really Kevin Herold, who's been my longtime clinical colleague, working with this drug almost as long as I have, really conducted this study showing that you could take individuals who were at the precipice of developing what we call clinical type 1 diabetes, meaning that they need insulin to keep their blood sugar in check. And just before that inflection point where you destroyed enough beta cells to become insulin dependent, treated patients at that early time point before actual disease, and showed that again, just 2 weeks treatment with with the drug, literally 2 years later, over half the patients that otherwise would have developed disease hadn't developed the disease with a single 2-week treatment. It told us a number of really important things, not just about the drug and how to hopefully get across the finish line, but it told us about the immune system, which is that type 1 diabetes is probably like a lot of other autoimmune diseases that we live with every day, multiple sclerosis, lupus, where the immune system waxes and wanes. You get these sort of bursts of activity, and then it goes quiescent, and then bursts of activity. And if you can get in there when there's a burst of activity with a drug that'll cause this apoptosis, then you can eliminate a lot of the cells that would otherwise be destroying the beta cells. And I hope that leads us to understand in the future, as we get biomarkers, we can identify patients just when they're starting to have that burst of a response, and then go in there, target those cells. And so my aspiration for this drug is not only will you be able to treat patients before they've been diagnosed, but you'll also be able to treat patients successfully just after they're diagnosed and they still have islet cells left. And ultimately, although I'm not a big Harry Potter fan, so I hope Matthias is right, I think ultimately the idea that we can come up with a drug, whether it be whatever it's working on or what we're working on, to give at the time that we give these beta cells, so that we can wipe out the cells that otherwise would recognize those cells as foreign, might be a nice match for a combination therapy. Because again, going back to cancer just for a quick second, nobody would treat a cancer patient with one drug and hope to have 100% success. The idea ultimately is combination therapies, and getting those pieces of the puzzle right is important. But if you do, then I think you can get long-term survival of the beta cells that are there, the ones that you've put in. And I think drugs like teplizumab will at least inform this a great deal of science on how we get there.
Right, and can you just mention the PROTECT trial that I know is it's on hold now because of COVID, but it will be recruiting again, right?
Absolutely. As I mentioned, you need to have, in order for a drug like teplizumab to work, any immunoregulatory drug to work, you have to have cells that still are present that can make insulin, because that's what you're preserving. You're preserving the cells that are remaining after autoimmune attack. And the PROTECT study is basically to treat patients with teplizumab just after you've been diagnosed. So within the first 6 weeks to 12 weeks after diagnosis, get in there and give the drug so that you can preserve the islets everybody knows about the honeymoon period. Well, the honeymoon period is largely a time in which you still have beta cells functioning, which help to control your blood sugar over that year or so after diagnosis. And if we can get in there quickly with a drug like teplizumab and shut down the immune system from causing more beta cell death, we can have an effect. So PROTECT is really for patients that are just at diagnosis, patients that can clearly benefit from shutting down the immune response for that window in which otherwise they would be destroying beta cells.
And we hope to get up and going and recruiting for that trial again soon.
We do.
So I don't know if you've been hearing the dings, but I've been getting a lot of questions from the audience. So I just wanted to ask one more question before we turn to the Q&A. I know we haven't talked about COVID, but I know COVID is very much on the top of mind for everybody in the world right now. And just for our last question, I wanted to ask you to make some ties between the COVID research, because there's definitely an immune response there, and how that might be benefiting from the research that has already occurred in type 1, and just how its effect on your research efforts in general.
Everett, why don't you start?
Yeah, I think I let my immunologist take the first step at this point.
Sure. Well, certainly COVID has changed medicine utterly. If you go into the hospital nowadays, you can't bring someone in with you. Everyone is masked and gowned. It's actually a much more safe environment now than ever before, I think. But it's certainly halted a lot of our efforts in terms of the clinical trial work. We don't have labs currently running. But you know, it is interesting how science works, because there are ways in which the science done through JDRF and others have helped COVID patients. For example, when patients get very sick in the ICUs, they lose a lot of ability to control their own blood sugar because of all the drugs they're getting, and their islets are actually not doing as well in general. They're sick as well. And in our ICUs, we're actually using remote sensor technology developed for T1D for patients who are in isolated rooms where going in and out is very difficult, so they can achieve better insulin control and hopefully better survival. So that's a really good case example of how fields you didn't think would meet do meet and inform each other. We have a lot of challenges. It's definitely important to keep the clinical trial infrastructure going. My son has been in a clinical trial at Stanford using devices, and it is possible to participate in clinical trials. That trial has continued. And so, you know, although it's making it much more difficult, we will find ways.
So, as Everett just said, we clearly, like everyone else, have been affected by the COVID crisis. There's just no way around this. So in San Francisco at the UCSF, most of our research labs are down. We have something we call a skeleton crew. Now, we and others are allowed to maintain specific experiments that are ongoing and that are long-term experiments. So it's not that all of our work has stopped, but the ongoing every day-to-day work did essentially go to rest right now. Now, we're using this time, of course, to be at home and having Zoom calls and meetings with members of my lab as well as collaborators all the time to essentially look at the data that we already were able to gather before we had to shut down and to prepare ourselves to move forward. Now, I think we've learned a couple of things from the COVID crisis. And again, I'm not talking about immunology here. Jeff can speak to this, and Albert already did much more eloquently. But in terms of what we've learned, I think the first thing is that data and science matters. That just being able to have accurate information from trusted sources makes a real difference for our society. And I think I certainly have to thank the JDRF and all of our funding organizations for the many years that they've supported us to get to this point. And we see that this is now moving forward. So stem cells, for example, human stem cells were identified in 96, 97. So they have been around for, less than the drug that Jeff was talking about. But what we have moved over these 20, 25 years or so towards actually making this into a hope for cell therapy, this is just unbelievable how fast this has gone. And everything was based on data and information and essentially trust in the work of others. So this is something that I don't think will go away, and I think we're going to increase and we're going to accelerate, because I believe many of our fellow citizens will now also, they just got reminded that having investments in science infrastructure, and specifically in life sciences, is something that saves lives in the long term. Now, to give you a specific example, I just told you we're making beta cells, and I know it's pretty hard to see this when I have my hands because they go in and out, but you know, a stem cell is here, a beta cell is over here. But in the process of making a beta cell, we're getting to a junction where we can also make something that would turn into the duodenum or the small intestine. Why is that important? Well, we do know that 25% or so of COVID patients actually have gastrointestinal disease as well, that we don't fully understand. So we already have written a protocol how we could use the information that, again, others, not my lab, but others, friends of mine around the world have gathered to use very similar differentiation conditions and now make gut cells out of this. So we can actually study COVID. So all of the work that has gone into just making beta cells already has provided tremendous amount of information to use this for other things as well.
So I'll end with just a couple of sort of scientific notes from wearing my researcher hat. I believe that the average person involved in understanding type 1 diabetes sees many hallmarks of the disease when they look at what happened with COVID patients, whether it's the implication that we have in type 1 diabetes that viruses may play a role in somehow triggering or exacerbating the disease, whether it's the same proteins, what we call cytokines, which are causing some of the damage in the lung that we already know causes some of the damage in the pancreas as a consequence of autoimmune disease, or whether it's frankly the blood glucose and the related regulatory pathways that control metabolism that are connected between COVID, as Everett said, and the disease. So it's not hard to go from a hunch that there's connectivity here to some real science and some real experiments that'll go in both directions. We've talked that both Everett and I are very interested in the cell therapy field of regulatory T cells. We're starting a trial in COVID-19, putting in those regulatory T cells to see if we can shut down inflammation in the lung, because that's, of course, not so unlike inflammation in the pancreas. We know that we can think about some of the other therapeutics that are being developed to try to treat autoimmune disease and how we might use those to treat some of the inflammatory aspects of it. And so I believe at the end of the day, that is, and it's a horrible situation. And of course, our hearts go out to everybody who's affected by this disease. That, to Matthias's point, if we gather information, gather data, gather facts in an effective way, I have no doubt that just like much of the work that we've been doing in immunology over the last 2 decades will impact on how we think about treating COVID-19, the reciprocal will be true as well. At the end of the day, we will learn some things about how this virus interacts with the body and the immune system at the body, which will give us some clues to how to go back and see if we can tap into that to help shut down some of the unwanted immunity that happens in type 1.
I think as you said, Jeff, that it's all one immune system, and what you've already unearthed with your work and with all of your work will definitely inform and feed into the work for COVID. So thank you. We have received a whole bunch of questions for you, so I think I'll try to turn to that. We only have about 15 minutes. But Everett, there have been several questions about your work, particularly with the islet transplantation work. And I might just combine several. They're questions just around how does it work if you actually can put your protocol in place in terms of making sure that the autoimmune attack doesn't happen again? You're dealing with the anti-rejection problem, but you still have the autoimmune attack going on. And also, people are asking, when can we start? When do you think that you will be able to start clinical trials with people with type 1? Are these phase 1 or phase 2 trials? So a lot of interest in your work.
That's great to hear. So it is a very complex problem, especially with type 1 diabetes and autoimmunity layered on what's known as alloimmunity, or the transplant responses to new tissue. There are mouse models that suggest when you take animals that are genetically different enough from animals susceptible to diabetes, and you transplant those islets in, it's a whole new immunological landscape. So when you induce tolerance to those new islets, the cells that may have recognized the old islets can't really jump over and be as active with the new islets. So there are different ways in which the tolerance mechanisms that we're using clinically could be extended to prevent T1D autoimmune responses. At least it's been shown in animal studies this is possible, but this is what we need to test. Certainly the data from the existing transplant patients, some of them who can go for many, many years with immunosuppression, suggests that the autoimmune component can be stopped to some degree, but the drugs that patients are on can have their own set of sometimes major problems. In terms of our efforts to translate, we're hoping, and with considerable help from UCSF and other centers like University of Miami, to be in trials with our kidney protocol in the next couple years. We're writing protocols together with leaders in the islet field, and we're already in the process of seeking approval. There's a huge infrastructure and a huge retasking that needs to occur. And we've built and brought together in an organic way, thanks in part, again, to the Center of Excellence, a big multidisciplinary team to put this forward, but a team that extends across institutions. And that's the way it's going to happen. So these trials will be phase 1 for certain in the beginning. And again, we hope that we'll be in trials very soon. There are other centers who are trying this approach as well, including City of Hope and Northwestern. We're actually collaborating directly with Northwestern to integrate in the Tregs that Jeff talked about into this whole protocol to see if they might be used in combination therapy in trials. That trial is actually open and accruing for kidney patients. We're hoping, even under COVID circumstances, to treat our first patient in May. So it's an exciting time, a lot of work. And the key is it's multidisciplinary, large teams. It's not one person, but many committed individuals working together.
Well, you should know there's a ton of interest and support in the community for your work, just based on the number of questions I've received. So thank you. Maybe the next question, Matthias, there was a question about Ips cells and being able to differentiate them not only into beta cells, but into alpha cells. And maybe if you can talk about that a little bit, and just the benefit of actually taking an Ips cell from an individual and using that same line to go back into that same individual.
Yeah. No, absolutely. So IPS lines stands for induced pluripotent stem cells. They are not being derived from the embryo. That was one of the hallmark discoveries that we've had over the last 15 or so years, done by Shinya Yamanaka, who is affiliated with UCSF and the Gladstone, but also has an institute in Tokyo. And the idea here is that you can take cells from somatic tissue. Somatic tissue means from someone who is an adult or a child, from the skin, for example, or from the blood, and then use specific factors to more or less move the cells back in time so that they become stem cell-like. That's why we call them induced pluripotent or stem cells. And these cells have the benefit of potentially coming from one person, then being isolated from that person, being generated into the cell that you want, and then retransplanted into that same person. So therefore, in theory, you should have a very little immune response. The problem unfortunately with type 1 diabetes is that we are generating the one cell that the body has decided to destroy. So even if you would take your own cells, the autoimmune disease against the beta cells would wipe out the beta cells when we put them back in. So that is the problem of doing it from a patient. Now, the IPS cells are very, very good and they're getting better. We're learning, again, more and more about how to generate them. And not surprisingly, if they come from a younger patient, they actually are more robust in its stem cell properties than if they come from someone of my age, for example, over 50. And therefore, people are now going out and actually generating whole libraries of these IPS lines with the intent of essentially having those that can be matched like organs can be matched to donors upon transplantation. So this is a really exciting area of work. And yes, they are a little bit less malleable than the embryonic stem cells still, but they're getting much, much better. And we can also, like with embryonic stem cells, in theory, generate every single cell out of this. And we are actually moving towards using induced pluripotent stem cells to generate these alpha cells and these delta cells in the islets that work together in conjunction with our beta cells to ensure that we release insulin only when it is appropriate. So yes, these cells will work.
Great, thank you. There are a couple questions around, what exactly are autoantibodies? How do they work? And I think also a connection with the HLAs that we do know give more of a tendency towards developing type 1. I don't know if Jeff, you want to start by maybe giving a little bit more of a explanation of how these two pieces of the disease work?
I'll give it a shot. So the immune system has multiple arms to it. There are two major arms to the immune system. One is your white blood T cells, which go out and seek cells that are different from self and destroy them. And the other are B cells, white blood derived B cells, which don't directly destroy infected cells or beta cells, but recognize and make antibodies that do the job of going and binding to those cells. So type 1 diabetes is no different than many diseases, whether it's virus infections or other autoimmune diseases, where you have both a T cell response and you have a B cell response. So you have T cells that go and directly attack the beta cells, and you have antibodies made by B cells that are recognizing antigens and proteins on the beta cells. What makes diabetes in one class of autoimmune disease is that the antibodies themselves actually aren't particularly dangerous. It's the T cells that do most of the damage, not the antibodies. The antibodies actually provide a flag. They're kind of what we call a biomarker. When the immune system gets activated and you make antibodies, then you can monitor that. And so patients that are at risk for type 1 diabetes are monitored by monitoring the presence of autoantibodies. But the autoantibodies themselves are not responsible for the destruction of the beta cell. The destruction of the beta cell is by the T cell. And so how does the T cell do that? It does it because it recognizes a flag on the beta cell as being unique, and therefore the T cell receptor, which is this receptor lock key, it's a lock key mechanism, that the receptor recognizes the key. And the key is a combination of your HLA, your MHC, your anchor protein that anchors peptides on it to be recognized. That's why when you get a flu vaccine, they put in the flu protein, and then your MHC binds it, and your T cells go in and recognize it. So that's what caused the disease. So it's not surprising that these flags, these proteins on the beta cell, aren't all identical in all people, because it's the structure that's created by the protein binding to the MHC molecule. And it turns out that in most autoimmune diseases, including type 1 diabetes, that certain people with certain MHCs, HLAs, are more susceptible to the disease, because think of it as a hot dog bun. They have a hot dog bun sitting on the surface of the beta cell that binds to certain hot dogs. And it's that hot dog, which happens to be insulin peptide in this case, that the T cells can recognize. So when we think about autoimmune disease, we think about T cells recognize, not B cells, they're the marker. T cells recognizing the hot dog bun and the hot dog here being insulin. And not every hot dog bun is the same. And so as we think about determining who's at high risk for the disease, we look at autoantibodies, but by then you already have started it. We look at the genetics, and most prominently the HLA, because that tells us who's at risk most because they have the right hot dog bun. And then we look at other genes like the insulin gene itself to tell us whether or not it's most likely that insulin protein will sit in the hot dog bun. Maybe made no sense, but that's the way I think about it.
Yeah, that was a great kind of 2-minute explanation of basic immunology. So Matthias, we got a question about encapsulation and other protective means to protect the beta cells.
Yeah.
And so I thought maybe you would want to talk about that. They specifically called out ViaCyte, but also, you know, you had mentioned gene editing, and that might
Yes.
actually be a way to skip over the whole encapsulation or their protective device type of approach.
Yes. Okay. And this is only going to take 20 minutes, so please settle down. Encapsulation. Why did we go into encapsulation? The general idea of encapsulation is, of course, is a fantastic one, which is we are generating these cells out of stem cells. We think they are safe, but who knows? There might be something that is not quite 100% safe. So we're going to put them in a bag. That's essentially is an encapsulation device. So that encapsulation device then does two things. It first protects the person who gets the transplant, because the cells in the bag cannot get out. And then second, it protects the cells within the bag from any kind of immune system that the transplant person essentially would level against the cells in the bag. So it's fantastic. The problem with that is that we have tried this for many, many years. And there's lots of really smart people and bioengineers, including lots of people at UCSF who are working on this, and we're getting better and better materials, better composition, better kind of architecture of the devices. So there's a lot of work that is going behind this. But at the end of the day, we still have the struggle of that the cells are sitting between the cells and the supply of oxygen, for example, and nutrients. There is a barrier, and that barrier often is not good for the cells. So if I go back to the ViaCyte trial, then the ViaCyte first trial was with an encapsulation device that was a closed device, and the cells, unfortunately, sitting inside the bag didn't do very well, likely because it didn't get enough oxygen and nutrients. And this can happen over the first week or two, and then the cells are not doing well, they actually die. Now, in the second trial, they punched holes in it, and therefore they had access to the endothelial cells that are coming in. These are the blood vessels, and therefore those cells actually were doing better. Still not perfect, but clearly better than in the first trial. So what I'm trying to say is that, in my belief, is at some point we're going to go away from encapsulation. I fully understand why people would like to do it, because you can put cells in there, you can keep them safe, and you can protect the person who receives the transplant. But that is not going to be an option that is available for almost every other cell therapy out there. If you need a cardiomyocyte, a cell that goes into your heart after you had an infarction there, you cannot put this in an encapsulation device because it has to integrate into the heart. The same is true for a dopaminergic neuron that goes into your brain to help you when you have Parkinson's. And so there's a tremendous amount of effort around the world with lots of stem cell people to try to figure out how either we cloak the cells that we put in so the immune system does not recognize them, or, as Jeff was saying, trying to modulate the immune system in a way that they are accepted and therefore as being recognized as our own. So I think encapsulation is something that is amazing, and if it works, it's great. In the long run, mid to long run, I think we're going to have something that is more a naked cell that does not require encapsulation. But that's a personal belief. I think you're muted, Karen.
Sorry, thank you. I was just thanking you all. I just got a notice that we are over time, and I've just noticed that we indeed are. So I am going to wrap up, and I am truly sorry we didn't get to all the questions. There were many, so there's a ton of interest in your work and the information that you are sharing with us as panelists. I just wanted to wrap up. There were several questions, excuse me, about clinical trials. And just mention that on the JDRF website, there is a really easy tool to find clinical trials and that you can actually search very easily by location, by proximity to where you live, by disease, by where you are in the, your experience with the disease. So I would encourage everyone listening to take advantage of that tool. And I think, there's very strong clinics at both Stanford and UCSF at both your institutions, where the clinicians are also extremely knowledgeable about the clinical trials that are available. And so you can always reach out to your particular medical care provider to find out more about that. And I definitely wanted to thank the community for the interest in participating in clinical trials, because none of this research could progress without these clinical trials. And so your participation is key. So thank you for that interest. And I'm not sure if the organizers are going to try to circle back to answer some of the questions that came online. But, I just wanted to express extreme gratitude for the three of you for giving up time on your Sunday to, again, support and educate the Type 1 community. And thank you to everybody who's behind the scenes running this summit. This session at least went entirely smoothly, and I greatly appreciate it. I don't know who's following up. Crystal, maybe, to close this up? Oh, you're muted, Crystal.
All right, I'm unmuted now. Thank you so much, Karen, for moderating. And thank you, Dr. Bluestone, Dr. Hebrock, and Dr. Meyer for your time and expertise today. This was such a great session with so many questions and engagement. Thank you all.
Thank you, guys. It was fantastic to be with you guys. Thank you.
Okay, good luck.
Thank you.